Well, welcome to the second lecture in the postgraduate lecture series. So the interventions that have truly had a major impact on human health for the most part really relate to infectious diseases. So you've got sort of vaccinations at number one or number two, and then antibiotics. And something that we often forget about is just basic hygiene and sanitation, and also living standards. And unfortunately, that's also something that really highlights the inequality in global health. And one of the bugs that also highlights this inequality is Streptococcus, and amongst the strep pyogenes or group A strep. So it's really a great pleasure to have Professor Mark Walker down from Queensland to talk to us about group A strep or gas. He's really made some incredibly significant and critically fundamental discoveries regarding the virulence factors and some of the mechanisms that changes strep pyogenes from being just a skin infection to a highly invasive and destructive organism. And this organism also has quite a spectrum of disease, from sort of a skin infection, pharyngitis or infection of the throat, through to glomerulonephritis, and also rheumatic fever. So Mark actually is the director of the Australian Infectious Diseases Research Centre at U of Q. And as I said, he's had a preeminent career in a lot of work regarding strep pyogeny. So it's a great pleasure again to welcome you, and thanks for the lecture.
Well, I'd like to thank you all for turning up today, and also thank you very much for the invitation. Because this is a postgraduate seminar series, I'm going to change tack a little bit and adopt a more narrative style, and hopefully give some insight how some of the things that we've discovered, and I should use parentheses there, it's really stumbled over, how we stumbled over a couple of new things in this particular area, and what we've been doing in the last 10 years since we started working on group A strep. Everyone in this room will have been suffered a group A strep infection at some time or another. Everyone's had a sore throat. Usually that's treated with strepsils and not much else. If you have repeated pharyngitis, you can go to the doctor, you can get treated with antibiotics, and generally things are okay and work out fine. Group A strep does cause skin infections such as impetigo. This is not particularly pretty to look at, particularly if it winds up on your face. However, this is not a life-threatening disease, and it can be controlled once again with antibiotics. We're very lucky that strep pyogenes is resistant to very few antibiotics and can still be treated with penicillin. And penicillin resistance has never occurred in this bug. Oops, going the wrong way. What we were particularly interested in is how come this bug, which is host adapted, it doesn't lurk anywhere else in the environment except in the human host, usually sore throat or no symptoms whatever, why in some instances does it go on and cause diseases such as necrotizing fasciitis or the flesh-eating disease? Why does it cause bacteremia? Why does it cause toxic shock? In a small number of patients, but for those patients clinically very important, necrotizing fasciitis and toxic shock, you can lose between 25 to 50% of those patients even with treatment. And the treatment for these diseases is very crude: amputation of the limbs, debridement of the dead flesh around the wound, using antibiotics, of course, to kill the bugs, but by that stage things are too late. So why does group A strep cause this disease? And that's principally been the focus of our lab in the last 5 or 6 years. It also causes autoimmune complications such as rheumatic fever and rheumatic heart disease, post-streptococcal glomerulonephritis, kidney failure, and these are autoimmune diseases. And I was quite interested to find that some people at WEHI are starting to get interested in acute rheumatic fever and looking at that. But I won't be talking any more about those particular suite of diseases today. As Mark mentioned, Group A strep is an important human pathogen. It ranks in the top 10 of infectious disease killers around the world, not quite as many as HIV, TB and malaria, but nonetheless a significant human pathogen, despite being sensitive to most antibiotics that are used clinically. The clinical epidemiology is represented in this particular slide. This is a pulse field gel electrophoresis where the DNA from individual clinical isolates is extracted, cut with a restriction enzymes and run out on a gel. And you can see that there are two blocks of bugs, M3 over here and M1T1 over here, that are highlighted. They cause about between 30 to 40% of infections in Western countries, such as Europe, North America, urbanised Australia. And the M1T1 strain really only popped up on the scene in the mid-1980s in the Rocky Mountain region of the US. The M1T1 strain, which is the bug that I'm focused on examining, has been genome sequenced, and it was found that it contains two bacteriophage in the genome. One bacteriophage encodes a superantigen called SpeB, which cross-binds T cell receptors and leads to an inflammatory response. SDA1 is a DNase, which bacteriologists call a spreading factor. And we'll be talking more about the role of the DNase in Group A strep infections during this seminar. The epidemic, once again, as Mark mentioned, that in lower socioeconomic backgrounds, Group A strep is much more of a problem. It's a particular problem in the top end of Australia in Aboriginal communities. And there's some interesting differences that epidemiologically, that no one's got to the bottom of. That in those populations, Group A strep is much more a skin pathogen, whereas in Western populations it's much more of a throat pathogen or pharyngitis-causing organism. The epidemiology is quite different, where there's over 100 circulating strains and there's no dominant clones such as M1T1 or M3. And this is another pulse field gel, just selecting a random set of clinical isolates from the Northern Territory, and you can see that there doesn't seem to be any particular relationships between the strains that are circulating. So epidemiology is quite different. Where did we start with Group A strep invasive disease? Well, we were interested in how Group A strep interacts with the human protein plasminogen. Now, I should say that this wasn't a great insight on our part. There happened to be a plasminogen lab next to mine in Wollongong when I was there, and we thought that it'd be quite nice to collaborate and have a look at that. So just a few notes on plasminogen. It's a human serine protease. It's involved in fibrin degradation, connective tissue degradation, basement membranes, etc. It is cleaved to form the active protease plasmin, so it's a zymogen. These kringles, numbered 1 to 5, are involved in binding to receptors. Generally, it's a lysine-dependent binding. And Group A strep is known to interact with human plasminogen. It has plasminogen receptors on its surface, and it produces streptokinase. And streptokinase is an enzyme that's able to activate human plasminogen. It was used as the treatment for heart attack victims prior to TPA, for instance. So it's a blood thinner. So plasminogen in the human system is tightly regulated. You have activators TPA and UPA that activate the molecule, and a whole bunch of regulation layered over that molecule. There are plasminogen activator inhibitors and plasmin inhibitors as well. However, when streptokinase activates plasmin, it gets around all of these inhibitory steps. So you get unregulated plasmin activation when streptokinase and group A strep is involved. So we thought this should be involved in invasive infection. The hijacking of this human protease, it degrades fibrin, connective tissue, all of those things that seem to be involved in necrotizing fasciitis. This should be a reasonable molecule to work on. So I'll just step you through a number of the interactions between group A strep and the human plasminogen system. So plasminogen can be activated by human activators, TPA, UPA, and it's able to bind to the surface of group A strep through a number of well-characterised plasmin receptors. Group A strep produces streptokinase, which can bind to plasminogen to form an activating complex, and this activating complex can convert plasminogen to plasmin once again, which can then bind to the receptors on the bug's surface. There's also what's called a fibrinogen-dependent pathway, where streptokinase and plasminogen can form a trimolecular complex with fibrinogen, and that complex can then bind to the surface of the bug either through plasmin or fibrinogen receptors and activate new molecules of plasminogen. So this is all known biochemically. So the question is, the last thing that we need to look at is that group A strep also produce a protease of its own. It's a cysteine protease called SpeB, and SpeB is the other molecule that we'll be talking about. There was a mouse model developed in 2004 where a mouse was transgenically engineered to produce human plasminogen. And the reason that was done is because mouse plasminogen is not activated by streptokinase. So if you want a mouse model of group A strep infection, an invasive mouse model, you need to add human plasminogen. And this was done by making a transgenic mice. You can see at escalating doses, the transgenic mice survive less well than the wild-type C57 black mice. So this now represents a mouse model that can be used to study particular group A strep virulence determinants. So where did we start? What we did is we collaborate with the Menzies School of Health Research up in Darwin, and we got a whole bunch of group A streptococcal isolates that were from clinical origin, and we started measuring the interaction of these bugs with the plasminogen system. And this first result here is just looking at plasminogen binding to the surface of group A strep. And remember at the start I said that this M1 strain is clinically very important, and it's also responsible for a disproportionate amount of invasive disease. And of all of these isolates, we have one M1 strain isolated here with the arrow, and we disappointingly found that the M1 isolate isn't able to bind plasminogen. So we didn't think we were off to a particularly good start. And even when you add fibrinogen into this system to form the trimolecular complex with streptokinase, fibrinogen and plasminogen, this particular M1 isolate still was unable to bind plasminogen, where you can see here many other of these isolates are able to bind plasminogen efficiently. So the M1 isolate that's important for invasive disease doesn't bind plasminogen. The other next thing we looked at, well, okay, what's happening with streptokinase production? And disappointingly, when we did a streptokinase assay, we found that the M1 isolate and a whole bunch of isolates over here fail to produce active streptokinase. This is a streptokinase assay which the readout for is active plasmin. We have other isolates over here, once again, that are able to produce streptokinase. And then the last assay we did was, okay, let's try to look at something that mimics a little bit more what's happening in vivo. And so we took human plasma, we grew the bugs in it for 3 hours at 37 degrees, harvested the bacteria and just measured plasmin activity on the cell surface. Once again, the M1 isolate over here isn't very good at sequestering plasmin and activating it on the surface when grown in human plasma. The final assay we did was we looked at this other protease called SpeB. SpeB is the cysteine protease that's produced by group A strep, and you can see many of these bugs produce SpeB, but some don't. And you can see it, well, at least this M1 isolate is capable of producing SpeB. So I don't know whether you're feeling particularly confused about how group A strep and this M1T1 isolate interacts with plasminogen and the plasmin system, but we were certainly confused. And this was an actual conclusion slide from a seminar I gave about 5 years ago. And after the seminar we did just wander over the uni bar and drown our sorrows because there seemed to be no interaction. And this is the same experiment where we've taken an M1T1 isolate, we've grown it in human plasma for 3 hours and measured plasmin activity on the cell surface. And at the time, the PhD student who was doing this, Fiona McKay, also threw in a mutant of group A strep that we'd constructed. And the mutant was a mutation in the cysteine protease SpeB. And when we looked at the same genetic background with an isogenic SpeB mutant, we see that now this identical strain minus the cysteine protease SpeB is able to sequester vast amounts of human plasmin on its cell surface. So at the time, we didn't really know what the hell does that mean. We seemed to think that that was quite significant, and our role next was to try and figure out what was going on. And lack of SpeB kept popping up in the international literature as clinically important. SpeB negative isolates, so this is just measuring SpeB activity in non-severe invasive infections and severe invasive infections. And there was this epidemiological link between SpeB negative and severe invasive disease. So that was kind of interesting for us. And I should say each of these dots represents a human invasive disease isolate where the strain has been picked out of the patient and then characterised, and probably about 25% of the dots over here didn't make it, just to keep things in perspective. We can't do those sorts of experiments, but when we infect mice, we can also measure the SpeB activity of the M1T1 isolate. And the M1T1 isolate we use is 5448, and it'll pop up again and again in the rest of this talk. But you can take your inoculum, pick out individual colonies and measure SpeB activity. And you can see here that the inoculum is uniformly SpeB positive. It produces the protease. If you put the inoculum in the skin of a mouse and wait 3 days, you can see that there are 2 populations that are formed. One that is still producing this protease SpeB and ones that are SpeB negative. And then if you look for the bacteria in the blood, the bacteria that have actually made their way into the bloodstream of these mice that are almost on their last legs are uniformly SpeB negative. So we hypothesised a mutation had occurred at the time and SpeB had been switched off. And then we could then use that in our model system and think, well, maybe the SpeB negative ones are now sequestering plasmid on their surface and are becoming more invasive. Okay, that's exactly what I said. So at the time we were thinking this, someone else was doing it, and they had also noticed this relationship between SpeB negative bugs and invasive disease, and they'd sequenced the genome of one of these SpeB negative isolates from a mouse that was originally SpeB positive. To cut a long story short, what was occurring were mutations in a regulator called COV-RS. COV-RS in group A strep stands for control of virulence, and it's a two-component regulatory system. You find mutations in COV-R, or usually truncations in COV-RS, in COV-S, and COV-RS is an important regulator of group A strep virulence, and it regulates SpeB production. And you can see down here, for instance, this particular M1T1 is from a human isolate. This is an M3 that we sequenced that was SpeB negative from a human isolate. Down here we have a strain which was passaged through a mouse, contains the same mutation. And over here, the 548AP is a strain that we chose to characterise. It has a one base pair addition, which leads to a truncation of COV-S. So mutations were linked with switching off of SpeB expression. Now that mutation that results in switching off of SpeB expression, we were able to show that SpeB degrades plasminogen, plasmin, streptokinase, fibrinogen receptors, and therefore, if group A strep produces SpeB, it knocks down the interaction between group A strep and the plasminogen system. When you knock out SpeB, then that interaction occurs. And so what happens in the mouse model, which is expressing human plasminogen, we have the wild type over here in green at 3.2 by 10 to the 8 CFU subcutaneously. You can see that about 50% of the mice are killed. However, if you take this COV-RS mutant, one base pair addition in COV-S, at half the dose, you get a much more virulent bug. So we hypothesise that it's the interaction with human plasminogen that's actually causing these group A strep to be virulent, and it's the mutation in COV-S or COV-RS that is allowing that interaction to occur in vivo. Now, this isn't a regulatory system where the genes are switched on and switched off. These mutations never revert back to wild type. So something's going on in vivo selecting for a COV-RS mutation, and then that's fixed in the population. So we wanted to know what the selection pressure for the SpeB loss was. So 6 years ago, I was actually on a Fulbright scholarship and in a lab that was doing microarrays. Here's the only microarray slide I ever did. There it is. We got one result out of it, that this COV-RS mutation affects about 400 genes, or 15% of the genome of group A strep. But we looked at the results of this, and you can see that the COV-RS mutation downregulates SpeB, which we were quite interested in because of the link with plasminogen. And of all this long list of virulence determinants that were upregulated, at the time we were particularly interested in streptodornase. The streptodornase is the DNA that's encoded by the bacteriophage that is found in the M1T1 clone. So we thought, okay, let's ignore doing microarrays further and just start looking at the interaction between SpeB and streptodornase. And we came up with this hypothesis using these furry creatures that you can only buy in the US. This is the benign group A strep. You can tell because it doesn't have the knife and fork down the side of its body to suggest that it's the flesh-eating form of the bug. The whole thing pivots on this regulator, and I've got these things in my office if you want to come and have a look one day. But anyway, COV-RS is the regulator. There are mutations that are occurring that are being selected for in vivo. SpeB is being switched off. The lack of SpeB means surface plasminogen is being upregulated on the bug. And the other thing that goes up is the DNase SDA1. And we thought it's the interaction between these three components that is causing the trigger for invasive disease. And we were interested in the DNase because in 2004, it was published that neutrophils produce neutrophil extracellular traps, where bundles of DNA, extra DNA, are squirted out into the environment with histones and other components to catch infectious disease agents and to help kill them. So neutrophils are one of the first line of defence in an invasive, in any sort of microbial infection. Here's a picture of some of my neutrophils squirting my own DNA over some group A strep that are in this mixed culture, and the DNA has just been fluorescently labelled here with, I think it was acridine orange, but I forget. Here we have an apoptotic neutrophil, but these ones are sending out DNA nets because they're in contact with group A streptococci. And we thought, well, maybe the upregulation of the DNase is having a selective pressure on the bug because it's allowing group A strep to escape from these neutrophil nets. And so we started to try and mine our way through that hypothesis. Here we have a bright field of neutrophils and their interaction with group A strep. So we don't put too many group A strep in here because if you do, then you just get a raft of DNA and you can't see very much. But you can see that these neutrophils are firing neutrophil extracellular traps out to try and trap the group A strep in the culture. These bugs up here are the COV-RS mutant that has an upregulated DNase, and when you do the identical experiment, you don't see any DNA nets because the DNase breaks down the DNA nets and then helps prevent the neutrophils from killing the group A strep. And that seems to us like good selection pressure. And so we can quantify the killing of group A strep in the presence of human neutrophils. Here we have the wild-type bug. About 70% of the population is wiped out in the presence of human neutrophils in 1 hour. Using the same multiplicity of infection, you can see that the COV-RS form of the bacterium, that increased expression of DNase, completely saves this population. In fact, it's above 100% because the control is the absence of neutrophils. So the bacteria growing better when you put human neutrophils into the system in comparison to when they're absent, and that's quite reproducible. So the bug, with its changed regulatory system because of this mutation and increased DNase production, does much better when neutrophils are around. So we would suggest neutrophils are the selective pressure here. So I can say that, but we need to go out and try and prove that. And the assay that we came up with was the following. We take a mouse, we inject group A strep in subcutaneously, we wait 3 days, a lesion forms that we can then cut out. So we're looking at the in vivo population. We can plate those out onto blood plates, take single colonies, put them on a milk plate that contains casein, And SpeB, the protease, is quite happy to break down casein, and you get this classic halo around the patched out colony, and a SpeB negative bacterium looks like this. So you can determine whether the population that's been passaged through a mouse for 3 days is SpeB positive or SpeB negative. And when you do that, each of these dots here, and measure the percentage that are SpeB negative, you can take the wild-type bacterium. Each dot is 100 colonies from a single mouse, and you can see, based on the classic genetic experiments of when mutations arise randomly, some mice have large populations that are SpeB negative and others nil, but you get this effect where there is switching from a SpeB positive to a SpeB negative state in vivo. If we knock out the DNAs, then the population retains SpeB activity. So if you take the DNAs out of the system, there is no selection pressure to become a COV-RS mutant, which is SpeB negative. If you pop the DNAs back into that background, then you get this SpeB switching starting to occur again. So selection pressure in vivo is abolished when you remove the DNAs that we know is capable of breaking down neutrophil extracellular traps. And additionally, if we take a historic M1 isolate that's not part of the M1T1 clone, that doesn't contain the DNase SDA1, then it also doesn't switch in vivo. So the selection pressure on the M1T1 clone is provided by the DNAs that came into this clone from a bacteriophage. So this allows us to put together a hypothesis where we have an infection coming in via the skin, perhaps passed from the throat to the skin to the site of infection where an invasive disease occurs. These bugs are SpeB positive, they produce SpeB, neutrophils come in and start killing the bacteria via neutrophil extracellular traps. The neutrophils then are exerting selection pressure on the population. A random mutation occurs in COV-R or COV-S, and those bacteria are now resistant to neutrophils because they have upregulated DNAs, and they're able to degrade neutrophil extracellular traps more efficiently. And one of the unintended, or we would hypothesise unintended byproducts of this mutation is also the increased interaction with human plasminogen. So now these bugs are able to coat themselves with human plasmin, activate plasminogen to plasmin via streptokinase, and go on and cause an invasive infection. Okay, so a number of questions should arise from this hypothesis, one of which is, well, why aren't all group A streptococci COV-RS mutants? If they're more resistant in vivo, why aren't they dominating the global population completely? And we thought about this ourselves and come up with the idea, well, maybe these bugs are not transmissible. Even though they're more resistant to neutrophils, they're better able to cause an invasive infection that particular genotype of group A strep can't transmit through the population. And what we've done here is just looked at some basic adherence invasion assays, the things that we think might be involved in transmission, comparing the wild-type bug in orange with the COV-RS mutant in purple. And you can see that the wild-type bug is able to bind fibronectin and collagen, and the COV-RS mutant is unable to do so. So it may be defective in its capacity to adhere. It's also defective in its capacity to bind to HEP2 cells and HCAT cells, so hopefully a pharyngitis or perhaps a HeLa cell line, depending on your point of view, and a skin cell line. So the wild-type bug can adhere to those cells, the COV-RS mutant is unable to adhere. The wild-type bug is better able to form a microbial biofilm than the COV-RS mutant. And finally, we have developed a skin adherence assay where we simply take a mouse, apply some Nair to get rid of the hair, and then determine how able the bacteria is able to bind to mouse skin. The PhD student volunteered to do it to himself, but there were a few ethical issues that we couldn't overcome. So we looked at the wild-type bug is able to bind to mouse skin, and the COV-RS mutant isn't. So it looks like this particular bug is non-transmissible, or a non-transmissible sort of dead-end type, but most unfortunate to patients with invasive infections. So the final question that I'll just address in the last 5 to 10 minutes is what happens in the non-M1 group A strep. We know that M1T1 contains this DNase, but not all the other group A strep. So what goes on there? This whole bunch of bacteria over here, which is also capable of causing disease. Are there COV-RS mutations occurring in invasive infections there? And the answer's pretty much yes. If you take non-M1 group A strep, you can find examples of bacteria that have lost the capacity to produce SpeB. And they've lost the capacity to produce SpeB because they've accumulated mutations in COV-RS. And those COV-RS mutants, these three here, are resistant, or these two here, and the M1T1 isolate over here, are resistant to human neutrophils. The wild-type bugs, M1T1 and these two non-M1s, are sensitive to human neutrophils. We can look at how the bacteria are able to switch in mice, and the big difference is that there's selection pressure on the wild-type bug to become SpeB negative that we just looked at. These non-M1 types that don't have the DNase are less able or unable to switch to the SpeB negative phenotype in vivo. The mutation still occurs, it's just not selected for. The wild type, which is able to form a COV-RS form in vivo, is able to kill human plasminogen mice. These bugs that remain wild type are unable to cause death in the same mouse line. So, but some of those non-M1 isolates were COV-RS mutants, so what do they do? So here we have the M1T1 isolate, and the two non-M1s are still highly virulent in the mouse model. So even though they're less able to switch to a SpeB negative state, we think that when they do, they're just as virulent as an M1T1 isolate. So to explore that, what we did is we took a wild-type bug that was non-M1T1, a bug that was a COV-RS mutant, we repaired COV-S so it produces SpeB, and SpeB is produced, capsule production goes down, and then we made the complemented mutant where we mutated COV-RS again, and you can see SpeB is switched off, capsule goes back up. Neutrophil resistance is high in the wild-type COV-RS mutant and the complemented mutant, but not the repaired strain. Plasmin accumulation is high in the CoV-RS mutant, the complemented mutant, and not the repaired strain. And the question we wanted to ask now is, we've repaired this bug that isn't usually, it was from an invasive human infection, but epidemiologically it's not particularly linked. Is it able to switch to a CoV-RS form? So we did the experiment and we saw that it was only rarely able to come to this form. So the mutation occurs, but it's not selected for in vivo. But when the mutation does occur, it's able to cause invasive disease because plasmin's accumulated on the surface and so on. And this bug doesn't contain the DNAs. So finally, what happens when you pop them into the mouse model? The CoV-RS mutant is virulent, the CoV-RS restored is completely avirulent, and the complemented mutant, once again, is virulent. So what does that allow us to conclude? Firstly, that mutations in CoV-RS increase virulence and are clinically associated with human invasive disease for this M1T1 type, which is the most important form of group A strep circulating in Western populations. That in vivo human neutrophils provide selection pressure for neutrophil-resistant M1T1s to occur. And this CoV-RS form, though it's resistant to neutrophils, has a colonisation defect, so it doesn't dominate the population. So neutrophil-resistant CoV-RS forms also occur in non-M1T1 serotypes, but may arise only infrequently in comparison to M1T1, and that's suggested by our mouse studies, and it's also supported in the literature epidemiologically. And so non-M1T1 serotype group A strep that do contain these CoV-RS mutants are more virulent in this mouse model. So it just leaves me to thank the collaborators that I had with this work. Peter, Jason and Andrew all went through and got PhDs out of this particular project. From the University of Michigan, David Ginsberg's an expert in human plasminogen, and his group, in particular Hongmin Sun, developed this transgenic mouse model. When I was at UOW, Fiona was the first person to start on this work and developed all the disappointing results, at least the results that we initially thought were disappointing, that grew into this insight on how group A strep causes invasive disease. And we work with people in the US at Tennessee and San Diego. So thanks very much for your attention.
You've talked a lot about the initiation of disease, and it's fairly easy to see how spreading will occur as a result of all of these mechanisms. But what do you think actually causes the tissue damage? And what's the next step? Why does the mouse die, and why do you get necrosis of the fascia?
So I'll separate those two things. The mouse is dying because of a systemic infection. And what we see that occurs in mice is after you infect subcutaneously, there's a fibrin deposition that is laid down around the site of infection. And in the mouse model, what we believe is occurring is it's the degradation of that sort of innate immune response and allowing the bacteria to go systemic that is causing disease in mice and killing them, usually between day 3 and day 5. In humans, it's not that simple. So what I think is occurring is 99.9 times out of 100, everyone is exposed to group A streptococci either in their throat and on their skin. And in the skin, you can imagine how that there'll be a normal innate immune response, and that clears the infection and you don't get necrotising fasciitis. When that does occur, though, it's clearly degradation of extracellular matrix components that allows the bug to get a foothold. The CoverS form of the bacterium, there's about 400 genes that are upregulated. Most of them are virulence determinants, and so that are overcoming complement degradation, complement deposition, overcoming different aspects of the immune system. So all of that's occurring. You've got plasmin being produced in an unregulated manner, and plasmid itself can trigger other matrix metalloproteins and so forth. But what I'm doing is skirting around the exact answer to what's going on in the fascia, because no one really knows. The infection in necrotising fasciitis is a very swift infection. It can be treated with antibiotics, but usually to no avail. And the other complication is that in some cases of necrotising fasciitis, there's no visible entry point for the infection. Sometimes there's a blunt trauma, but it's hard to imagine how the bacteria are gaining access to the internal layers of skin and into the fascia if there's no obvious entry point. There has been a hypothesis that perhaps bacteria are hitching a ride in macrophages or other white blood cells and arriving at this point of trauma and gaining a foothold there, but no one really knows. There's been talk of using plasmin inhibitors as treatment for necrotising fasciitis, because even in the best medical care, the prognosis is not particularly good. So the answer is no one really knows the exact mechanism beyond, the stage of initiation.
I'm just wondering about, they need to be able to, they get a mutation in the COV-S system,
Yep.
which then makes them virulent, which then leads to a virulent phenotype. But then you say that those bacteria then cannot pass on to another host, or they're not as able to do that, it's readily able to do that. So it means that getting a mutation there is advantageous, but only in that particular host.
Yes. So I think there's a hierarchy here where I would say that every group A strep has some capacity to cause invasive infection. Then group A strep that have a CoVRS mutation have a higher capacity to cause invasive infection. And if the bacterium is prone to, or has a system where there's selection pressure that drags a population of CoVRS mutants out of the general population, then that particular clone, such as the M1T1, and M3 also have a similar bacteriophage, may be at the top of the tree in their capacity to cause invasive disease. But the bacterium, so that's a hierarchy, but from the bacterial perspective, clinicians and people involved in health are focused on invasive infections because they kill patients. From the bacterial perspective, the bacterium doesn't want to kill the patient, it just wants to transmit. So we believe that a CoVRS mutant is a dead end, that it's unable to transmit, therefore you never see build-up of these mutants in the group A strep population, but if there's selection pressure to drag those mutants out, then mutations occur randomly, and random mutation occurs in that locus, and it's selected for.
Do you think they have
Yes.
being able to mutate?
Yes, because they have the bacteriophage encoded DNA such that a mutation in COV-RS increases expression of that DNase and makes it more resistant to neutrophils in comparison to the wild type.
So leading on from that, how do the non-M1T1 bacteria that don't have the DNase that do then become invasive survive with neutrophils if they don't have the DNA?
We're studying that at the moment, actually. So we know that a COV-RS mutant that is in a non-M1T1 background is also resistant to neutrophils. And that in that background, there is no DNase, and we find that when you mix those bugs with human neutrophils, there's plenty of DNA nets that are present, but the bacteria are not killed by them. And we've actually done a screen, and we see that there are a couple other virulence determinants that may be involved in oxidative stress resistance that are important for resistance to human neutrophils. So we've got that far, but there doesn't seem to be the strong selection pressure where a COV-RS mutant is selected out of the population in a way that the M1T1 is. So the same concepts are happening, but there are different resistance mechanisms that are going on.
Do the DNA nets actually catch them, or they avoid the nets?
The DNA nets are not broken down, is what we can say, and the bacteria seem to be resistant to the capacity of those neutrophils to kill the bugs.
Slightly off topic question. How does this relate to what scabies or scabies mite, which kind of parasites can infect?
So it's interesting that in the Northern Territory Aboriginal populations, that group A strep is much more associated with skin disease such as pyoderma, and the clinical people up in Menzies and at other places have hypothesised that the group A strep in those populations is being transmitted on the backs of scabies mites. And so it may be that for that group A streptococcal population, there's clearly major differences in how they're transmitted. It doesn't seem that group A strep causes throat infection so much as skin infections, and it may be the transmission is really wholly dependent on the presence of scabies mites in those human populations.
So do you think the scabies might harbor those viruses and bacteria?
Yeah, so this isn't my work, but some of the clinical people in Menzies would say that the group A strep in those populations is getting around via scabies mites, and they've generated some data to suggest that. But it's certainly very difficult to find, and in those populations there's high levels of invasive infections, probably one of the highest recorded levels of invasive disease caused by group A strep. But the transmission cycle doesn't seem to be from pharyngitis to pharyngitis. It seems to be skin to skin, and scabies may well be involved there.
Did you have a question, Justin?
Well, I can do it very, very quickly, I guess, thank you. With the COV-S mutant, I just wonder if it can rescue the non-COV-S mutant. So you've got DNase producing bacteria protecting those nearby bacteria that can't produce those DNases, and your model kind of got me thinking about that. So have you done the different experiments where you mix different ratios and then
Yeah, yes, we did that, and we didn't see we didn't see a rescuing of the phenotype, at least in terms of disease causation. That when we thought that if we seeded a wild type population with a COV-RS mutant, that we'd probably get a more rapid progression to invasive disease and death, at least in the mouse model, but we didn't see that. It seemed to follow the same time course.
So if you enumerate the numbers though, so you pull them out of the mouse and then do your casein assay,
Yep.
or your milk assay, and actually count, do they let's say that the COV-S or the DNA is non-producing bacteria, did they die out completely, or are they able to persist because of the DNA is producing bacteria?
Well, we can generally only measure persistence over 3 to 4 days, because after that the mice tend to either recover or pass away. But we started looking at that but didn't progress far enough to allow me to answer your very intelligent question.
So we've got lunch outside for people that would like to ask some more questions, so Mark will be around for a bit longer. Thank you very much again.